My name is Hana Zelená. I'm from Institute of Public Health from the Czech Republic, from Ostrava, and I'm the head of biological department of our institute. And thank you very much for invitation to this workshop. And I'm going to present here our results about evaluation of several diagnostic methods for detection of SARS-CoV-2 infections. So next slide, please. Next, okay. So you can see at this slide, you can see the most commonly used diagnostic methods for detection of SARS-CoV-2 infection. And most common and most widely used is definitely RT-PCR method, which is highly specific. It's a relatively rapid method. And the question is, what do these respective methods in fact detect? So what's the target of the detection of these methods? So the RT-PCR doesn't detect the whole virus, but it detects specific sequences of RNA, which are specific for this virus. The RT-PCR, thanks to its high sensitivity, is considered as gold standard. And as you see in this picture, this method detects not the whole virus, but only its RNA, which is the carrier of the genetic information of the virus. Recently, there are also plenty of so-called rapid antigen tests, which are very simple because they don't need to have some specific laboratory equipment.
I don't hear you, Hana.
So then we have rapid antigen tests, which detect not the RNA, but they detect specific structural proteins, which are the part of the structure of the viral particle. Most commonly, the target protein is nucleoprotein, or it also can be spike protein or other structural protein on the surface of the virus. The rapid antigen tests are less sensitive than RT-PCR, but on the other hand, they are very simple and very fast because the result is available within 15 minutes. So immediately at the place of the test, you can have the result. And the third method, which is not intended for routine use, but better for confirmation, is the virus cultivation, also called viability test, which is the only method. With this method, we can detect the complete infectious virions. This method is relatively highly sensitive, but it detects not the RNA molecules, but it detects virions, direct the infectious viral particles, which means that only in case that we have the positive test of viral cultivation, we can be sure that the investigated sample really contains infection particles and that the tested person is infectious. Next slide, please. It is important to know the quantification of the respective structures of the virus. It is interesting to know that one infected cell, which is infected by the SARS coronavirus 2, produces in average 10 to 100,000 molecules of RNA, but this number of RNA molecules corresponds only to 10 to 100 complete viral particles, which means that RNA molecules are within the infected cell produced in very huge excess, which is 1 from 1,000 to 10,000 more than is the number of complete viral particles. It is a very important message because it is important to know this when we somehow want to evaluate our methods. There is also another term, so-called infective dose, which is the number of viral particles which are able to infect the target human being or susceptible cell culture. When we are talking about infective dose in laboratory conditions, which means when we infect the susceptible cell culture, there is missing any immune response, any protection by immunity, and therefore theoretically even one complete viral particle is able to infect this cell culture. But in a living organism, like in a human or in a susceptible animal, there is also fighting against the infection immune system. And that means that it is necessary usually that the viral infected dose has to be higher than in laboratory conditions. So for susceptible person, the minimum infective dose is at least 100 or 1,000, but for some persons, even more viral particles are able to cause the infection in human body. So this is a difference. So next slide, please. So as I was talking before, the most commonly used method, the most widely used method for detection of infection is PCR, or in RNA viruses, we are talking about RT-PCR, which means reverse transcription PCR. As you probably know, normally for PCR, we are working with the structure of DNA. But in case that we are investigating RNA viruses, the pre-step of PCR is so-called reverse transcription, which means that we have to transform RNA to DNA, and then it continues with the DNA. So, and each PCR reaction consists of several so-called temperature cycles, which is the cycle consisting of 3 steps. In the first step, we have to heat the mixture to high temperature, which is higher than 90 degrees of Celsius, which causes denaturation of the double-stranded DNA, which is divided into 2 single-stranded DNA. Then comes the second step, so-called annealing, in lower temperature, when the temperature goes down to from 50 to 65 degrees. And in this step, the primers are bind to the complementary DNA strand, which is called hybridization. And then comes the third step, so-called elongation, when the new DNA strand is synthesized with the help of thermostable polymerase. And then is created the DNA strand, which is complementary to the DNA template. So, and these 3 steps are repeatedly repeated. And therefore, the name of the reaction is the polymerase chain reaction. Next picture, please. So you can see it here in this picture. The first step is the denaturation, then is the hybridization or annealing, and the third step is the prolongation. And usually we repeat these cycles up to 40 times, and in each of the cycle, the number of target DNA is doubled, which means that when we have only 1 single copy of DNA in the beginning of the reaction, then at the cycle 30, we have 1 billion of DNA copies, and in the circle 39, we have 550 billions of DNA copies. So here you can see the extreme multiplication of only one copy of DNA which is present in the beginning, and the extreme sensitivity of this method. So next slide, please. Then what is also important to know the terms which are commonly used for quantification of viruses or RNA copies. There is quite often you can hear the term so-called viral load. One can think that it means that it is the number of viruses, but in fact, the viral load means that it is the number of RNA copies, not the viruses. When you want to describe the number of viruses, you have to talk about infective dose, which really corresponds to the number of infectious virions. As we have seen in the previous slides, that the number of viruses is much more high. The number of viruses is much more lower than the number of the RNA molecules. So next slide, please. Then the second method, which is commonly used for the detection or for the infection control, are antigen tests. They are very fast and simple, so it is very easy to perform it, even by the people which are not used to work in laboratory. And these tests, these antigen tests, detect structural viral proteins. They are based on the binding of antigen with antibody, and then we can see specific color reaction. In the case of antigen tests, we don't perform any amplification, and therefore this test is much more, the sensitivity of it is much more lower. But what is important, that the sensitivity of antigen tests very greatly varies depending on the manufacturer and also by the examined sample. There are tens or even hundreds of various assays for detection of antigen of various manufacturers, and the quality of them is very variable. From antigen tests of very high quality, very good sensitivity and specificity, but there are also available plenty of assays for detection of antibodies, antigens, which are of a very poor quality, as you could see in the next slide. Next one, please. So this is the typical picture of this this antigen test. Next one, please. So in this graph, you can see what are the advantages or also disadvantages of those both methods for detection of infection. In this line, you can see the time. So the infectious virus is present in this time. Relatively short time, the infected patients produces infectious viruses. But when you use very highly specific PCR, you can have the positivity before, but also sometimes very long after the infection ends. And when you use very highly sensitive method, which is PCR, you almost always detect the infectious person, but you also very often have positive results, even in case when the patient is not infected anymore. On the other hand, when you use test with a lower analytic sensitivity, typically antigen test, there is some risk that in the phase of in the phase when the number of viruses is very low, that this antigen test can miss the infectious person. This period is relatively low, but the period when the patient is really very strongly positive, very infectious, usually the antigen test with good performance are able to detect infectivity of this person. So next one, please. Yes, and So, and then is the third method for virus detection, which is the virus cultivation. The cultivation of virus is very specific and special method. Not each laboratory is able to do it. And most problem of it is that it takes quite a long time, and it is necessary to work in a special conditions in the laboratory with high containment, so-called BSL-3 laboratory. We perform this viability test in latent tubes, which contain sensitive or susceptible cell culture from monkey kidneys, CV1. Then we add to each tube some small amount of the sample from nasopharynx. Then we incubate this mixture in the thermostat at 37 degrees at 5% of carbon dioxide. And then we have to examine each of the tube every day under the light microscope in relatively low magnification of 100 or 200 times. And we search for typical cytopathic effect, which is caused by the infection of the virus, because the virus changes to the infected cell changes its shape, and we can see the typical changes which is caused by this virus. And once we detect cytopathic effect, then we made the next stop, the confirmation either by electron microscopy or by PCR. This method in laboratory conditions is very sensitive. We are able to detect very low amount of infectious particles present in the sample. Next one, please. So as you can see, because the SARS-CoV-2 virus belongs to the group of highly dangerous pathogens, we have to work with these living viruses in a specific laboratory with high containment laboratory called BSL-3. It's because of the safety to the personnel. Next one. And here you can see the latent tubes containing the cell culture with the virus and the thermostat when the cultivation is done. Next one. Here is the light microscope. We have to examine the tubes every day in this microscope. Next time. And this is the picture we can see in the microscope. This is the cell culture, and in the middle of the picture, you can see the typical changing of the shape of the infected cells, which we call cytopathic effect. And once we see cytopathic effect, then we used electron microscope to visualize the viral particles. Next one, please. And this is the picture of SARS coronaviruses in the magnification of 100 to 200,000 times. And in this huge magnification in electron microscope, we can see the typical viral particles of coronaviruses. Next time. So, and now our results. We evaluated 10 different antigen tests from various manufacturers. 6 of them were done from nasopharyngeal swabs. 3 tests were from saliva samples, and 1 was from nasal swab. The patients which were investigated were outpatients, which means that there were people which were either asymptomatic or only mildly symptomatic patients, which were tested either due to typical symptoms for COVID-19 infection, or there were contacts to positive persons. And we tested the people by this antigen test, and at the same time, we performed another swab from nasopharynx for a standard PCR test. Then we compared the results of those 2 methods, and those which were discrepant, either PCR positive antigen negative or antigen positive PCR negative, we verified these results by virus cultivation as you could see at the previous slides. We tested altogether almost 3,000 persons. Of those, approximately 1/3 were positive in PCR test. Next one. And here it is the first result. In this slide, you can see the results of sensitivity. You can see 10 tests, 6 of them from nasopharynx, 3 from saliva, 1 were from nasal swab. And in the column number 3, you can see the declared values of sensitivity of those tests which were declared by the manufacturers. As you can see, the values of sensitivity was pretty nice, declared by the manufacturers. But when we compared our real results from our patients to PCR and viability, the results were not as good as was declared. Only 3 of all the tests, the sensitivity was sufficient enough, but in 7 tests, the sensitivity was lower than is admired by ECDC, because according to ECDC, the minimal required sensitivity for antigen test should be 90%. And 7 of the tests which were evaluated in our laboratory performed worse than 90%, although the declared sensitivity was perfect in all of them. Next one. This is the same for specificity. And this is the same. All the tests declared very good specificity, more than 97%. But in our evaluation, 6 of them had specificity sufficient, but 4 of them had specificity lower than is required by ECDC. Next one. And here is the final evaluation of both sensitivity and specificity. And as you can see, from 10 tested antigen tests, only 3 succeeded, and their minimal sensitivity and specificity were sufficient for the according to the requirements of ECDC, and the other 7 failed. So they shouldn't be used in Europe. But when you consider only declared values, all of them are normally used in our population. So this is the big problem, that the real quality is much more worse than declared. Next one. So this is also very interesting, another result of our study. Next, please. Yeah, there are some circles. Yeah, and you can see that altogether about 600 samples had discrepant results, and of those, only 85 were false positive in antigen test, but more than 500 PCR positive were negative in antigen. But when we compare it to viability, then you can see that only less than 1/2 of the samples contained infected virus. And all samples with viability less than more than 34 contained no viruses. Next one. So the last message is that the antigen tests have very different quality despite of the perfect parameters declared by manufacturers, and therefore the independent evaluation is needed. And another important, that sufficient results had only three tests which were taken from nasopharynx. And another important message that is that viable virus, which means infected virus, was present only in 44% of the antigen, so-called false negative, compared to PCR only. And the vast majority is of weakly positive samples contain no viable viruses. And on the other hand, the well-performing antigen test can represent valid tool for infection control in high prevalence settings, but not in low prevalence settings, because it is important to take into consideration the low positive predictive value in low prevalence settings. And the important thing that the test of viability is the only way to detect if a sample contains really infectious viruses. So the viability should be considered real golden standard. So next one. Thank you for your attention, and that's all. Thank you very much.